Tanis. I'm a third-year assistant professor in biological sciences at UD, and today I'm going to tell you about what we were able to do on a specific project during my INBRE pilot grant, and how we discovered that C. elegans release distinct populations of extracellular vesicles from neurons. And I'm talking here about exosomes and microvesicles. But I have to say, when I started the INBRE pilot, I don't even think I knew what an extracellular vesicle was. So how did we get here? Well, my lab studies a gene called calcium homeostasis modulator 1, and it is associated with acceleration of Alzheimer's disease. So it was originally identified as a gene of unknown function. It's expressed in the hippocampus and located in an AD linkage region. A specific variant, the P86L polymorphism, is associated with the acceleration of Alzheimer's disease onset, and it decreases CALM1 mediated permeability. So at this point, it was hypothesized that CALM1 could be an ion channel. And loss of CALM1 actually causes an increase in the toxic A-beta species in the brains of mice. But to this point, the actual physiological significance of CALM1 in the mammalian brain remains unclear. So through the work of my own work and the work of others in the Foskett lab, we did show that these CALM proteins are indeed ion channels. For ion channels, they're relatively non-selective. They're permeable to calcium, sodium, potassium, and chloride. Their gating is regulated by voltage and extracellular calcium. And they have a wide pore diameter of about 14 angstroms, which might explain the relatively weak selectivity. And importantly, CALM channels have a unique pharmacological profile and have no sequence similarity with known ion channels, indicating that they belong to a new family of ion channels. Now, there are 6 CALM genes in humans, but the model organism that I use, C. elegans, has a single CALM homolog that is 16% identical to human CALM1. But importantly, CALM1 in humans and C. elegans have similar biophysical properties that I just outlined previously, and they're functionally conserved. So the CALM1 mutant in C. elegans, if we express human CALM1, it can actually rescue the mutant phenotype. So while the human brain is very complex, the C. elegans nervous system only has 302 neurons, and so we decided to use this simple model to figure out what CALM1 is actually doing in neurons. So the first thing we needed to do was figure out which neurons specifically CALM1 is expressed in. And so to do this, we created a transcriptional reporter consisting of the CALM1 promoter and regulatory region driving GFP expression. And here you can see all of these neurons in green. That indicates neurons where CALM1 is expressed. And we co-express this with a transcriptional reporter with the CLIP6 regulatory regions driving mCherry. And we know the neurons that CLIP6 is expressed in, and so by looking at the overlap, we were able to identify the specific neurons that CALM was expressed in. And we found, these are images of the male head and tail, that CALM is expressed in some male-specific neurons called the CEMs, the HOB, and the RNB, as well as a few other ciliated sensory neurons that are found in both males and hermaphrodites. We then used a functional fusion protein to determine where CALM1 localizes in these neurons. And so in the 2 images on the left, we used that CLIP6 promoter to drive GFP and fill out the neurons. And we also expressed CALM1 tagged with TD tomato. And we found that it localized to the cilia of these sensory neurons. And then to confirm this, we co-expressed a functional tagged PKD2 GFP with CALM1 TD tomato and saw that they co-localized. All right, so CALM1 localizes to the cilia of sensory neurons. What do we know about these sensory neurons? Well, the only thing we really know is that extracellular vesicles are released from these neurons. So again, I'm showing here the male head and the male tail, and extracellular vesicles are released from the ciliary base into a lumen, and then they are released out into the extracellular environment. So here's PKD2. This is a trip channel, which is a known EV cargo. And tagging this with GFP, you can see all of these vesicles are released. Now what I have labeled as the male tail, that just appears green due to cuticular autofluorescence. So what are extracellular vesicles and why are they important? Well, they play an important role in intercellular signaling. They transfer bioactive molecules between cells, proteins, mRNA, microRNA, lipids. And interestingly, different subpopulations can be released from the same cell. So this suggests that there must be a cargo sorting mechanism that allows for certain cargo to be sorted into one subset of extracellular vesicle and a different cargo to be sorted into another subset. There are different types of extracellular vesicles that are largely categorized based on their mechanism of biogenesis. So exosomes are released when multivesicular bodies fuse with the plasma membrane, whereas microvesicles form when the plasma membrane buds. And it is now appreciated that EVs play key roles in development and homeostasis, as well as progression of neurodegenerative diseases and cancer. So it's really important that we understand how the basic cell biology of EV biogenesis and EV cargo sorting, because there really is disease relevance here. So our first hypothesis, well, CALM1 is permeable to calcium and it's permeable to ATP, and maybe CALM1 is required for the release of these EVs by, because the mechanisms of EV biogenesis often require calcium or ATP. So we cross the CALM1 mutant into this PKD2 GFP strain. But as you can see here, CALM1 is not required for the release of these extracellular vesicles. So we thought, well, it's localized up in the cilia of these neurons. Maybe CALM1 is an EV cargo. And indeed, we found that CALM1 is in these extracellular vesicles released from males and also from hermaphrodites. And we used 3 different types of imaging to determine this. We used Zeiss confocal with Aries scan detection, super resolution, and we also used spectral unmixing to show that these really are indeed GFP vesicles and not just some sort of background noise. I'd also like to point out that our CALM1 GFP is expressed at endogenous levels. So this is not an overexpression artifact. Our next question was, well, is CALM in the same subset of vesicles as PKD2? And so we made animals that expressed functional tagged CALM1 TD tomato and PKD2 tagged with GFP. And as you can see in the merge, these cargo appear to localize to different vesicles. Okay, you see essentially green vesicles and red vesicles, but no vesicles where the cargo overlap. And as a control, we express CALM1 TD tomato and CALM1 GFP in the same worms. And here you can see they overlap in all the vesicles, as expected. So since they are in different vesicle subsets, we wanted to know if factors that were required for the release of the PKD2 vesicles were also required for the release of CLHM vesicles. And there are no known biogenesis factors, but this gene CLIP6 is required for the release of vesicles from the lumen into the extracellular space. And you can see here in the wild type, there are lots of PKD2 vesicles released, but in the CLIP6 knockout, this inhibits the release of vesicles. However, loss of CLIP6 does not inhibit the release of CLHM1 GFP vesicles, as you can see both in the images and in our quantitation over here. So this suggests that not only are there two distinct subpopulations, but they have different biogenesis and release mechanisms as well. So we are currently working on identifying genes that affect the biogenesis and cargo sorting. And we have one potential positive result so far with an associated protein, ALX1. And when we knock out ALX1, we see an increase in the number of CLHM1 GFP vesicles that are released. And this was actually surprising to us because the mammalian homologue of ALX1, Elix, actually plays the opposite role in biogenesis of extracellular vesicles. So we actually expected to see a decrease in release. So currently we're hypothesizing that it actually is instead playing a role in cargo sorting. And we are following up on this right now by crossing this mutant into the strain that labels both populations of vesicles to see if now there's more overlap in cargo. So why are we studying these EV subpopulations in C. elegans? Really, this INBRI pilot allowed us to set up this model to gain some preliminary data. I submitted an R01 in February that was scored, so I was really happy about that. I don't think it's in the fundable range, but I was just happy to get a score. So really we actually were able to set up this model during the pilot. And one big question in the field is how specific cargo are chosen. But in cell culture studies, these studies don't take into account the in vivo cellular environment. And for EVs being purified from tissues or bodily fluids, this results in heterogeneous populations of EVs. So it's hard to gain information on the cell of origin and cargo sorting. And so our model is really unique in that it allows us to visualize these different subpopulations and actually express these fluorescently labeled cargo at endogenous levels. So our big questions moving forward are to define fundamental differences between these populations. And we are looking at other potential cargo. Are there only two subpopulations? Are there more? Do the vesicles differ in their size or their morphology? What factors are required for their cargo sorting? We would like to identify factors that regulate the biogenesis of CLHM1 containing EVs. And we're taking both a candidate gene approach and a forward genetic screening approach. And we have already identified one gene, ANOH1. It's a lipid scramblase that affects biogenesis of CLHM1 containing EVs. So we believe that lipid asymmetry does play a role in this biogenesis. And finally, we'd like to identify additional physiologically relevant cargo in CLHM1 containing EVs. And we have now successfully tagged CLHM in two locations, a GFP on the C-terminus, which is intracellular, but we also have a strep tag in the second extracellular loop, which will allow us to pull down these extracellular vesicles and then perform mass spec to identify protein cargo. So I just have one other, the pilot, we actually worked on a whole bunch of things. I've only talked about one project here, but I would like to also take one slide to talk about a method that we developed in the lab, which I think has broad application to many people, not just those studying C. elegans. And we're currently writing up a paper on this. So, you know, a lot of us use, those of us who are doing genetics research, use mutants that have single nucleotide polymorphisms. And when we want to genotype these animals that have single nucleotide polymorphisms, unless it changes a restriction site, in order to genotype, we need to isolate the DNA and send it for sequencing. And this is labor intensive and also can be quite expensive because we do so many genetic crosses. And so Dennis in my lab wanted to develop a new tool to identify single nucleotide polymorphisms without having to perform either restriction digest or sequencing. And so we adapted super selective primers. So the premise is there's an anchor sequence that binds to the DNA, a bridge sequence, which is not complementary to the intervening sequence in the genomic DNA, and then a foot sequence, which is about 7 base pairs that anneals. And then the last nucleotide is the interrogating nucleotide, which can distinguish a mismatch between a mutant and the wild type. So the idea is that there's a common, we'll say, reverse primer, but then there's a primer that specifically identifies the wild type, or in this case, specifically identifies the LEV11 mutant. And as you can see in our gel here, the primer specific for the wild type across a wide range of annealing temperatures only identifies the wild type allele. And then the primer specific for the LEV11 allele only recognizes the LEV11 mutant. And so these super selective primers can be used for routine genotyping, screening strategies, now that we're doing a lot of CRISPR, and it can also be used for things like colony PCR to identify successful site-directed mutagenesis constructs. So if you're interested in this technique, feel free to talk to me more about it. And with that, I'd like to thank everyone in my lab, especially Mike, Dennis, Rachel, and Jackie, who've done the most work on the CALM project, Jeff Kaplan and Sylvan in the Bioimaging Center for all of their help because imaging these EVs has been very technically challenging. And Papani, who's helped us with some preliminary mass spec work, Matt Hudson for helpful discussions about EVs, and of course Delaware Embry for the funding that really has allowed us to make a lot of headway on multiple CALM projects in the last couple of years.